Can you imagine being able to edit the most basic and hereditary unit of life? DNA. This is precisely what CRISPR technology allows us to do. CRISPR gene editing is used extensively worldwide. But what exactly is CRISPR? Let's go through it in detail. CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. It is a gene editing technology that allows us to make precise changes in our DNA. Originally, CRISPR was discovered as a defense mechanism in bacteria in the late 1980s, where it serves to protect them from viral infections. During a viral infection, viruses enter the bacterial cells, where they take control of the host machinery. Then they use this factory to make thousands of copies of themselves. After this process is complete, they escape the host by breaking its cell wall, which ultimately kills the bacteria. Somehow, few bacteria survive this infection. They store a small piece of this virus's DNA. We call it a spacer. This DNA is then transferred to a special protein in bacteria called Cas9. Now, whenever the invader shows up again, this protein uses this information to scan this foreign particle. And if it indeed contains the sequence, then it cleaves the DNA at that sequence, making the harmful organism useless. Scientists used this mechanism to develop a gene editing technique known as CRISPR. But how did they do this? CRISPR technology works like a pair of molecular scissors that can cut DNA at the locations that we want. CRISPR mainly consists of two main components: a Cas9 enzyme and a guide RNA. Cas9 enzyme acts as a scissor to cut the DNA. But how does it know which part of the DNA to cut? For this purpose, it takes the help of guide RNA, which acts as a guide and directs Cas9 to the exact location in the genome. These guide RNAs can be easily made in labs and are also made by biotechnology companies that prepare specific RNA sequences according to the requirements of the scientists. To understand it better, consider a scenario where we want to cut a specific human gene. We will give a copy of this gene to Cas9. Now we will put this inside the human cells. It will go into the system, find that gene, and then cut it. Once the DNA is cut, the cell's natural repair mechanisms come into play. There are two primary repair pathways: homology-directed repair, HDR, and non-homologous end joining, NHEJ. Now, scientists can manipulate these repair processes to add or delete pieces of sequence in DNA. This is termed as gene knock-in and knockout. Let's first look at HDR. It is mostly used to knock in certain genetic modifications. In our body, there are two identical DNA molecules lying side by side. They are technically called sisters. The repair machine uses the information from this sister DNA to repair the damage in the DNA we cut. Scientists can modify this sister DNA molecule at the location where they want to make a change. So, when the repair machines copy the information from this DNA, it will help us to add the specific change that we want to add. On the other hand, NHEJ works differently. It rejoins the broken ends of DNA without requiring any sister DNA molecule. This can disrupt the function of the gene and hence is mostly used to knock out certain traits. To know about the limitations and ethical concerns related to CRISPR, check the article on our website.